Okay. 1 minute. Sì, sono lì. So, hello. I haven't slept since the Congress in Leiden decided to host the next edition here in Italy. And as I told the decision, Jonathan started losing sleep too. But here we are. We have a very dense program and a tight schedule. So I will start right with introducing the President Giovanni Laezza, President of the Fondazione Museo Civico di Rovereto, that is hosting us. And after will speak Jonathan Stanker, the head of the section of zoology. Please.
Good morning, and I'm sorry for my English. It's not very good, and I prepare a little speech. Dear participant, it is with pleasure that I welcome you to the Rovereto Civic Museum and to the 4th European Congress and Orthoptera Conservation. Our museum has a long history of activity dedicated to the study and valorization of the nature and culture of the Trentino Alto Adige region. In recent times, the conservation of biodiversity has become increasingly urgent, so I welcome to the proposal to host this European Congress on Conservation in a museum because it fits well with the activities of our institution. The conference poster is not accidental. It depicts Zoineriana marmorata, a cricket endemic to the coasts of the northern Adriatic, which was thought to be extinct. In 1996, Cloikers and Fontana found a surviving population, and this event stimulated the research that led to the identification of other populations. Our museum therefore launched a research project that led to the founding of a new population. What was done with eagles and lions, we have done with a species of cricket. We hope that this will serve not only to help nature in a time of crisis, but also to instill hope in scholars and the new generation who are facing to nature conservation. At the end of this little speech, I want to say many thanks to Mr. Filippo Maria Buzzetti and Mr. Jonathan Stanker. I ask one applause. Thank you. Thank you, many thanks, and good work to all.
Hello, good morning everyone, and welcome to the fourth edition of the European Conference of Orthoptera Conservation. Before to start, I would like to spend a few words to thank the people that made it possible. I sincerely thank Filippo Buzzetti. He proposed that the fourth edition of this Congress take place here in Rovereto. His dedication made it possible for this museum to host such an important conference. We are happy to have him as the museum main entomologist. This museum was founded more than 170 years ago, thanks to the commitment of some volunteers dedicated to the study of natural sciences, including entomology. After almost 30 years of inactivity, entomology has grown in the last few years thanks to the commitment of Filippo and myself, and thanks to the generosity of some researchers and donors. The collection have grown from 90,000 to 150,000 of specimens in the last few years. And we host about 200 of holotypes. All of the new specimens belongs to the Orthopteroids insects. And one of the donors is here with us, so thank you, Paolo Fontana. I want to conclude with the following consideration concerning animal conservation. Some studies have highlighted a significant bias in the conservation of animal species. When we have to choose which one to protect from the risk of extinction, we tend to focus our attention to the cutest one and to the species that are the most similar to us, systematically ignoring the less aesthetically species, such as insect, for example. But studies unequivocally demonstrated that animals that currently need to be protected the most are actually fish, amphibians, mollusks, and insects. But despite this, the conservation efforts are focused on the 0.2% of the animal species of this planet, that is, on mammals. Almost all the invertebrates are completely omitted. This is called the conservation bias. So we are trying today to place our small contribution to correct this imbalance. So thank you all and good work.
So thank you, Jonathan. Thank you, Mr. Giovanni, President. And now some practical information. For everything you need, please ask me or Jonathan. Then toilets. This floor right after the coffee room. Coffee break will not be in the coffee room, but will be downstairs in the garden. And as you know, this Congress is in presence, but we are streaming now, and each session will be recorded and available in the YouTube channel of the museum. Each session has one or two chairmen that will introduce the speakers. Visiting the museum will be free for you, but please wear the badge. Then, before the coffee break of today afternoon, we are planning a group photo that will be taken in the garden, downstairs. In the coffee room, there is a table with books published by the World Biodiversity Association, of which we are glad to have the president here, Mr. Paolo Fontana. And the posters are in the coffee room. We ran out of pins. We are going to buy some new pins. We will have lunch at Risto restaurant. Here you have the map, and as you can see, it is very near, just 7 minutes walking, but we will go together. You have in the bag, in the yellow bag, you have a ticket for the lunch of today and tomorrow, yes. And I will send you a form to vote for the next destination of the next edition of the European Congress. And so now some changes for today program. Landman presentation will be the last, not the second. And now start with Axel Hochkirch, the first chairman of today. He is president of the Orthopterist Society and co-chair of the IUCN Orthoptera Species Grasshopper Special Group. Thank you. Here you have the...
Okay, so thank you very much, Filippo, for organizing this Congress and for introducing me. And yeah, I hope we will really have a nice, I think we will have a very nice meeting here. So I will introduce the first speaker, which is Luc Willemse. Luc Willemse is probably well known as one of the co-organizers of the last European Congress. So he's based at the Naturalis in Leiden and an expert on Greek Orthoptera, as most of you probably know. So Luc.
Okay, thanks to the organization for allowing me to say a few words about field guides and field courses to stimulate the study of... Oh, sorry. I do it again. Thanks very much for allowing me to give this short presentation on field guides and field courses to stimulate the study of grasshoppers. So this is not really a research project I'm telling about, it's just some thoughts I had about trying to get some more people interested in this wonderful group of insects. I always wonder, I myself was raised in a family which more or less life was centered on grasshoppers, so in my case it's not really a surprise that I ended up as an orthopterist. But I always wonder when I meet people how each and every one of you ended up in this room, let's say, how your story is behind how you got interested in grasshoppers. So this lecture is about trying to find new people to interested in grasshoppers. So I'm going to give I have four short sections I'm going to talk about. First, I will talk about some challenges we are facing right now, not only about grasshoppers, but probably more in general about insects. Then I'm going to tell you a bit about the field guides and the field courses, mainly focused on Europe. And of course, Europe is not the whole world, so there's more in the world, especially if you look at biodiversity. Most of our biodiversity in the world is found in the tropics, so I'll take you a little bit also outside Europe to the tropics, and out of that I'll have some food for thought for all of you concerning the raising interest for the future generation of orthopterists. What we do as specialists, as researchers, we do research, and in the end we publish papers in all kinds of formats about a lot of topics. Here you see some which are more or less in the corner of taxonomy, phonistics, phylogenetics. And by doing so, we raise our knowledge about what we know about grasshoppers. This graph is just to illustrate. I needed a graph to illustrate that we know more and more and more with the growing years. So this is about the number of species we know from Greece, but you could also use a similar graph for the species of the world. They keep increasing, and they keep increasing because we do research and we found new species and we describe them. What can you do with this knowledge? Well, obviously you can, to start with, if you describe a species, you know that it exists, that's the first thing. And secondly, you know where it's found in the world, and you can create maps. For instance, this map is taken out of IUCN Red List Assessment for Europe, published some 7 years ago, and it more or less, this graph is this map is just based mainly on the number of species we know and their distribution in Europe, and it shows where you can find most of the species in Europe. Of course, we also do research on not only on taxonomy and phonistics, but on all kinds of other topics linked to grasshoppers, like ecology, the biology, etc., etc. By using this kind of knowledge, we can even start interpreting more things about grasshoppers. But still, there is, if you look in detail about what we really know about the European species of grasshoppers, for sure, taxonomy is quite well represented because we probably know 90%, maybe more, 95% of the species living in Europe we have been described, so we are very well organized there. Phonistics probably also quite, we know a lot about it, but then the other topics mentioned here are maybe a bit less well known, and maybe some of them we really know hardly anything about. It's also shown in this dashboard at the right-hand top corner. You see a gray area which represents species which are data deficient, which they call in IUCN if you don't have enough information to say whether or not a species is threatened, and if so, in what category it falls. So although we think we know a lot about European grasshoppers, I think I can state definitely that we still know a lot, we don't know a lot about grasshoppers. So the first challenge we have is we are lacking a lot of knowledge about the grasshoppers. Secondly, we are here in a room, a lot of you are doing something maybe with describing species or taxonomy-related topics, and you could say maybe you are also filled in the questionnaire Axel sent around some years back to ask what you're doing regarding taxonomy, if you are a taxonomist, etc., etc. These two dashboards show for Europe, for all the European countries, how well represented taxonomists are in the European countries, and whether or not they are threatened. I mean, it's maybe a strange way to look at it, but in a way, these dashboards indicate that if it would be completely green, we would be on the safe side. But no, it's not completely green. There are really some issues with taxonomists in Europe. We are lacking, in some countries, we are lacking quite a few taxonomists. In some countries, we are okay, but it's overall, it's not something to be, it's not really satisfactory. This is the same graph as these dashboards, but then you can see it in the country scale of Europe. Also, if you look more deeply in detail about the taxonomists in Europe, there's a lot of skewness in all kinds of stuff, in the gender, in the distribution, in experience, in age, etc., etc. So there is another challenge for us, and this one is about specifically from this report, it's about the Orthoptera. And I'm happy to say that although it states that the Netherlands are critically low, I think the main reason behind that, Axel, is that the Dutch Orthopteras didn't fill in the questionnaire. But okay, never mind. It's we'll try to do something about it. Don't worry. Okay, so the second challenge would be the taxonomist, the specialist, having enough of them. This third one, I just picked a picture from the internet, and that's the third option. The third challenge, of course, is our biodiversity, which is we are losing biodiversity, so that's also quite a challenge. Okay, I've stated the number of 3 challenges, and what can we do about it? Well, the answer is very simple. We need more specialists. We need more specialists. We need more specialists. Maybe not for the biodiversity loss. Maybe that's a bit exaggerated, but I think more specialists would help definitely to first fill in the gaps of knowledge and also to get some more people working on the stuff. There are various ways to do this. All have been you can capture on the outreach, either indirect via tools, social media, popular works like field guides, or you can do it directly in presentations, face-to-face with people, in media, in television, radio. You can give lectures on Orthoptera. You can give a course, a field course, or you can, if you are working at a university, you can have a student to work for you on something related to grasshoppers. Just two examples of indirect ways of raising interest in grasshoppers. These are online tools. One is a tool developed in Switzerland to identify the local fauna on the top left. The other one are also the observation tools which are on internet, which also somehow stimulate people to look at grasshoppers and raise interest in this group. Then, of course, what I'm mentioning in my title, field guides. Field guides are very nice ways of representing our knowledge about grasshoppers in a certain country in Europe. Here you see a number of examples from a number of countries. All these books have been published, I think, after 2005, so the last 20 years. It's quite good. If you look at the map of Europe, you still see some crosses in this map, which means, maybe I'm wrong. If I'm wrong, I'm sorry. But there's still some countries in Europe which do not have a field guide. And again, if you like to raise interest in other people for the Orthoptera, it would be very nice if, for your country, for your region, you do have a field guide, because it's a complete overview. You can illustrate it with very nice images. You can put keys in there. You can even, in the former days, you put a CD. These days, you put a QR code so people can listen to the sound. You can publish maps. So it's a very complete overview of what we know about, in this case, grasshoppers in a certain country. And mainly, another thing which is very important to tell you maybe is that if you have a field guide, it becomes much more easy to start thinking about giving a field course. Because if you give a field course and you do have a field guide, these two match ideally, and you can really have a good time to get a group of students interested and learn more about grasshoppers. These are two examples of field courses which have been given last year, one in the States and one in Greece. These are some images of the field course in Greece last year. I think the most important aspect of the field course is that you are interacting with the students. You can, if you are enthusiastic about your group, you can really, and you can bring it over to the students. It's a very good way of interesting students in this group. Then we go to the tropical areas. I'm going a bit fast. I'm running out of time. I'm going to tell you a little bit about the tropical area, because in Europe, things are not always looking as good as they could. But let's go to the tropical areas. Tropical areas, the south of the equator, the number of species there are, of course, many, many more than here in Europe. The number of undescribed taxa are a lot more than here. The number of researchers working there, working on, in this case, Orthoptera, are very, very few. There are very few collections where people can turn to if they want to compare stuff. There are no libraries, hardly any libraries, and online access usually is a lot more limited than here in the Western world, in Europe or the States. I take you to one of these tropical regions, which is called New Guinea. This is the map of New Guinea, and to the right you see the map of Holland, just to give you an idea how big New Guinea is. It's a very big island. It's a very different fauna. These are the crickets and bush crickets, some of them I saw last year when I was there. And these are some of the Silifera, which we saw there last year. Just to give you an idea about the necessity to start raising interest to work on the tropical fauna, there are two groups in this slide, one of Conocéphaline and one of Tetragidae, and it tells us how many species were known until recently. And for the Conocéphaline, Siegfried Ingwisch started working on them after 2000, and he published 173 new taxa just for this subfamily from New Guinea. The same applies to Tetragidae. Only 66 were known until 2000, and then when Joseph Stumbring started his study on the Tetragidae of West Papua, he described over the years 145 new species. So they're very rich areas. Okay, this one went wrong, but it's more or less a summary of the two slides before. The differences between the Western world, in this case Holland and New Guinea, are enormous in all kinds of ways. These are I reached the end of the presentation. So the food for thought I like just to present to you is that we are here gathered in this room. Some of you just started learning about or becoming interested in grasshoppers. Some of us are already doing this the whole life through. I think as a person, we could really once in a while sit back and think, okay, what else can I do except just publish research papers? Can I do something else to bring over my enthusiasm to other people, to young people, so that in the future, and let's say in 2050, when we have another ECOC somewhere in Europe, there will be an equally sized room, and maybe even there will be more people to listen to the presentations like this. And then when we talk about more general about the world, tropical areas, these are some questions we could ask ourselves about what we here in Europe, what is our responsibility in Europe or in the Western world when it comes to improving the backlogs in taxonomy and available taxonomic expertise in these tropical areas. Maybe we think it's their business, but maybe if we think we could do something about it, maybe we should. The other question we could ask ourselves, what is the importance of digitizing our collections, which consists for a lot of specimens from these tropical areas, as a way of repatriating this knowledge. Again, like for Greece or the USA, we can also think of developing a program, field courses in these areas to assist them in getting students to get to know grasshoppers. And then again, always the question starts at the end, who should start taking actions to do this? Should it be top-down? Should it be bottom-up? And if we want to do something, where do we start? How do we start? That was my presentation. Thank you.
Thank you, Luc. I think we can have one question, maybe. Is there a question or remark? Is it yours? Okay. Yeah. See.
So just one remark. There was marked that Moldova has a field guide, but I think it's that you put from Romania.
From Romania, yes.
Yeah, so it's not Moldova.
Yes, right.
Croatia doesn't have field guide as well. So,
Okay, there's still some more work to do, even. Yeah, we have gaps in Europe, but you're right, we have even larger gaps in the tropical countries where all the biodiversity is, so it's really important to build capacity there. And I hope we will see some movements in the next years to build more capacity in the tropics. Okay, I think we have to move on with the next presentation. As Filippo said, the presentation by Armin Landmann will be the last in the session, so the next one will be Oliver Havlicek. Well, he will talk about the conservation genomics of Gamsocles glabra in Western and Central Europe.
Yeah, thanks Axel, and thanks everyone. I'm glad to be back here, but back at the ECOC, and newly to Rovereto. Yeah. And very happy to see all of you guys again, or newly. Yeah, with my presentation is going to be a bit more specific than that of Luc, but I'm hoping to address some of the points that you raised with this project that we had here. Yeah, to introduce myself, I work at the University of Zurich. I'm a lecturer for biodiversity of invertebrates. I worked before in Hamburg, in Barcelona, and in Munich. And yeah, one thing of how we can address Luc's point is that in Zurich we started a new study program called Biodiversity, a bachelor's and master's that is meant to, among others, really train people as specialists. But I'll say a few words about that at the end. Yeah, about Gamsocles. To start, I want to look at the habitats. And the habitat that was most relevant for us there is steppe habitats. These are globally very large. There's a very large contiguous steppe zone in Eurasia and in Central Europe or most parts of Europe. There's just isolated patches, fragmented patches of this habitat that we'd like to call extrazonal patches. These are often threatened, but partly there's also areas that act as replacement habitats, such as military training areas that have a steppe-like vegetation and can host steppe species. There are some, like montana, that are typical for that, or Stenobotros crassipes, and our focus species is Gamsoclase, the heath bush cricket, which has the largest part of its distribution in the global contiguous steppe zone, but has isolated populations throughout central, western, and some other parts of Europe. And these are the ones we studied. There are more, to which I'll also come back, but the basic idea was to start studying the populations in Germany, and then we joined with the colleagues from the Netherlands, from Slovakia and from Hungary, and sampled some areas, such as this place in Netherlands, which is a military area that's under military use and habitat management. The same is true for the German sites, but in Slovakia, this is a xeric grassland that is now mostly used as a cattle pasture, and in Hungary, it's a pusta grassland. And all of these, are inhabited by Gamsoclase. And the questions we asked were, can the populations of these various habitat patches, first, can we distinguish them using population genomics? And then second, can we detect gene flow? That is, admixture in individuals or dispersal of individuals. So we wanted to know how isolated these populations are and how this is relevant for conservation. We tried to sample minimum invasively, so we just took one leg and then released the specimen. We were not able to control the viability of these specimens, but at least they jumped away, so they would still be active. And then we use DDRAD sequencing, double digest restriction site associated DNA sequencing, which is a method that is typically used for population genomics. So in very short, we use just a few words on the lab and protocol. So the DNA is digested by enzymes, then size selection is taking place. So this creates basically a subset of the whole genome of anonymous markers, but this is basically a method to work with species with whole large genomes, such as most Orthoptera, to still get a genome-wide sampling, but at a lower cost, so we could sequence more individuals at the same time for the same effort. And then this data is used computationally using the so-called STAX pipeline, which has the advantage that you don't need a reference genome. You can assemble de novo and extract all these SNPs, single nucleotide polymorphisms, so variant sites across the genome, and you can get around 20,000 in that case. And then using structure and structure harvester, we would test how many populations could be identified using these genetic methods, and then if these populations were admixed, so if there was gene flow between these various populations. And this is what it looked like. So you see that normally one color should represent one population. You see that the first Slovakian population is blue, light blue. Some of these are rather uniform. There is Germany 1 in the middle, which probably looks black to you, but is supposed to be dark violet. And then there's the orange one. Oops, that's probably supposed to be Germany 2. And then 3 on the right. So some of these are rather uniform, but then others are not, either are not uniform in themselves, or we find the same colors, so the same genotypes shared between these populations. So, yeah, I don't think I can easily point on that without leaving the microphone. It's okay. So you see that there is probably something like a mixture between, or using the mouse. Thank you, perfect. You see that there's genotypes shared between the populations of Slovakia and Hungary, and then, you also see admixtures, so you see some individuals that have like admixed parts. For example, here in that, what's Germany 2 in truth, from Germany 1 that is admixed. Also here we see some of this. These pink genotypes are admixed here. And then, also very interestingly, you see that in the Netherlands population, this looks like a big green block, but then there's also 2 individuals that are genetically fit much better with the German population here. So there is something going on. The question is, what is going on and how could it have happened? Natural dispersal, we don't know. So generally, the Gamsoclase glabra is able to fly and they can probably cross some distances, but then the distances between these different populations are pretty big. So we don't know if they are able to disperse on their own. Perhaps they did that. Perhaps historically the habitat connectivity was higher, but then even Hartz in 1957 wrote that Gamsoclase was restricted to some isolated patches of habitat, at least in the area he observed, so mostly Germany or Central Europe. We also see that proximity per se is not necessarily relevant. So you see that the 2 sites from Slovakia, 1 and 2, they are very close to each other, and there is some admixture, but basically not a lot of genotype sharing, much less than with the Hungarian population here, which is further away from this Slovakian population. Could it be human translocation? We don't know that either, but perhaps. So my favorite example is that of the Netherlands population that has some individuals that look more like the German ones. And this may be because these 2 military areas are part of the Dutch-German military collaboration. So maybe some of the Gamsoclase of the heath bush crickets hitched a ride on a tank or so. Yeah, some further observations, especially when we went to the Slovakian sites, we found, or we failed to find Gamsoclase on many of the sites that Tono Kristin, where he still recorded them in 2005 and 2006, published in 2007. So seems like in many, at least in this part of the range, the species is declining, and yeah, we should study it urgently. As you can imagine, this is just a very quick look into the species that has a much wider distribution. Also the sampling, you see that we had some good sampling for some of the sites, but only very few samples for others, so there is a lot of work that could still be done in that regard. And then obviously many areas were not yet sampled, so we didn't include any samples from France, from Austria, from Poland. We didn't include any from further east, from the real Eurasian steppe belt. And then there's populations that we don't know yet. So maybe some of you know that a new population of Gamsoclase was detected around Munich in 2022, just too late for us to include in this one, but this is something that we could do in a follow-up study. Yeah, that was my part on Gamsoclase. Just very quickly before I finish, I want to advertise a little bit our new biodiversity program. We started out last fall, in fall 2023, with the first cohort of bachelor students. We are very happy that we had 80 students in the major, so Hauptfach basically, and 80 minor who selected this. And yeah, we also have a lot of registrations for the next cohort, and we have many registrations for the masters, which is starting this fall of this year. Among others, we want to focus on, really we want to lay the foundation at least for people to become specialists in taxonomic groups. But we also have a focus on biodiversity and society, which also kind of docks on to that part of how can we motivate people to become interested in biodiversity, or how can we teach it to people. We have a big focus on fieldwork, and yeah, for the taxonomic knowledge, maybe you know that in Switzerland there's similar programs in other countries, but there is a certification system, and we will teach people so they can take these certificates and become official specialists of some groups. And that's it. Thanks for listening. I don't know if there's time for questions, so yeah, that's it from my side.
You stayed very well on time, so we have time for questions. Paolo.
Thank you, Oliver, for the presentation. I would ask if, by genetic point of view, there are data supporting that there was a refuge area in southern Europe, or this European population comes after the last ice stages from the east?
Yeah, good question. And in fact, we did not include enough samples from Far East enough. So I think we'll only be able to answer that when we have a broader sampling. The goal of this was initially just to see how distinct those isolated populations are from each other. But to answer the wider questions, we will also need a wider sampling.
Thomas.
Just one background information from the Austrian populations concerning this natural dispersion. Gamsa case was really critically endangered in Austria. Now it's expanding again, unfortunately. And we have singing males up to 60 kilometers away from the traditional core areas. So the natural dispersal seems to be quite a good factor for it.
Yeah, that's very cool information. Thanks.
Yeah, last question.
Can we estimate how long ago were those intermixing events? Basically, that those proportions of the genotypes differ vastly among populations?
Yeah, we didn't do any estimates of that yet, but I think with a bigger data set, it would probably be something. Yeah, thanks.
Okay, thank you very much, Oliver, for a nice presentation. And yeah, good to hear that Gamsoclase is expanding, and I didn't know this. And in fact, that was always a species of conservation concern in Europe, and maybe it is benefiting from the changing climate, or yeah. Okay, so we come to the next presentation, which will be by Lisa Reis. Lisa is my PhD student and also project leader of the project ELSA, which is a project for the conservation of Barbatistis sericauda in Germany. Lisa.
Thanks. Yeah, good morning or good day from my side. I'm very excited to talk about monitoring and habitats of Barbatistis sericauda in Germany today. Most of you know Barbatistis sericauda. It's one of the bush cricket species bound to woodland habitats. Nymphs hatch in the understory in April. Oh no, that doesn't work. Oh, don't matter. Yeah, nymphs hatch in the understory in spring, and they move up towards the canopy where they live as adults at approximately 10 meters height. And males sing in the ultrasonic range. And this can not only be amplified with an ultrasound detector, but also made visible with a very cool device we have at the university, which is an ultrasound camera. And I wanted to show you how this works, but I guess it does not. Oh, it does. Okay, you will see two males very soon here. In the canopy, singing their typical syllable song. But due to this overall hidden behavior, before 1980 there were only 2 documented observations in Rhineland-Palatinate, which is in southwest Germany and part of our project area. And since ultrasonic detectors were used more commonly in Orthoptera research, observations increased and the status changed from extremely rare to widespread but not common. So this is the current known range of Barbatistis sericauda according to the IUCN, and you can see our project area in this brighter and darker green. And as Axel already mentioned, he launched the project ELSA in 2021, which kind of translates to development of near-natural oak forests for the common saw-tailed bush cricket and other rare or endangered insect species. And it's a conservation project with the research part. And in the research part, we had following questions. What is the optimal monitoring method for Babetistes sericauda? Can we get a glimpse on population dynamics within the project term? Is there a preference for a specific forest type? Because sometimes a preference for oak hornbeam forests has been suggested. Or can the occurrence be linked to other habitat requirements, like a well-developed understory? And we also wanted to know what are the exact distribution patterns in the study area. So to test what is the optimal monitoring for Babetistes sericauda, we set up 9 transects in Rhineland-Palatinate in the project area, each 1 kilometer long, and we tested different methods. I will keep these short. Ultrasonic detectors are the best method to map and monitor Babetistes sericauda, but we monitored these transects along the whole summer, during the whole summer and during whole days. So we can, for 2022, we have this complete pattern, and what was very surprising for us is that the highest song activity was in 2022 in the beginning of July, directly in the beginning of July, and we saw a huge drop in the song activity after the first week of August already, which was very surprising to us because phenology is supposed to be, until September. The daily singing pattern looked like this throughout the year, basically, with the highest song activity from 4 to 9:30 p.m. So this is the best time to map this. On 5 transects, which were already defined in late 2021 after we started the project, we can compare the data of the transects. And what's really interesting here is that this maximum activity in the beginning of July with this huge drop in the first week of August or the beginning of August seems to be unique for 2022. And we also see that the number of singing males really accelerates in 2023. So we are very looking forward to collect more data this summer, the next, and the year after that, until the project ends in 2026 and 7. And we are hoping to find explanations for these patterns. Our hypothesis now is that this pattern we see throughout these 2 years has something to do with the extreme weather we had in the study area in 2022. It was the second hottest and driest since start of the records in southwest Germany, with an average temperature of 20 degrees, which is way over the long year average of 16.3. So what was also interesting to us is that we saw that most of the known occurrences of Babetistes sericauda are located in Natura 2000 areas in the study area. And this was a huge bonus. This can be an observer bias, of course, but this was a huge bonus for when thinking about habitat preferences and habitats, because in Natura 2000 areas, georeference data about protected habitat types is available. And in the project area, we have 7 protected forest habitat types. And we categorized them into 5 categories. First, beech forests, which tend to have a little understory. Then beech forests, which tend to have a more diverse or better developed understory. Then we had oak hornbeam forests as one category, teleoaterian forests as one, typically on slopes and ravines. We also included alluvial forests, and we included not protected forest habitat types. We got this data from forestry management, and also mapped spruce forests and non-classified mixed stands. And we conducted a randomized survey, evenly distributed along all these habitat types, on random points, with a 15-minute survey on each random point. And we counted the singing males, so most of the time we counted 0. In the end, 187 random points were available for data analysis. And the results look like this. As you can see, Babetistes sericauda occurs in all surveyed forest types, but this was kind of already known that it can occur in many different forest types. But we see no significant association with one of them, so we cannot support the hypothesis that there is a preference for one specific forest type. What we also did is on a pool of these random points, 50 plots, on 25 Babetistes sericata was present and on 25 Babetistes sericata was absent. We conducted a detailed habitat analysis. First, we analyzed the canopy structure and the light regime via hemispherical photography. You can see two of these hemispherical pictures on the left side of the slide. We did a Braun-Blanquet vegetation survey on each of the plots. It means a detailed analysis of all the plant species and their abundances on the site. And we estimated the vertical connectivity of the leaf area, kind of equivalent to a horizontal cover estimation. Just to give you a picture of what hemispherical picture analysis can do. After field work, we had 9 pictures which looked like that for each plot, and we used the software Hemisphere to analyze them. And this software gives you the following data. First, a value for the light transmission in a 2-dimensional projection from the zenith towards the ground. Also, a value for the vertical light transmission through the canopy. Then, next, a value for the canopy openness. It's also called the sky view factor, and which means basically what is the amount of sky you can see in a 3-dimensional projection when you are on the ground. And for each of these values, the amount of statistically significant large gaps, which means, okay, what is the amount of large gaps in canopy openness? And last but not least, it also calculates the leaf area index, means what is the leaf area when you would project all the leaves in the canopy towards the ground. Okay, of course, these are all highly correlated variables, typically for ecological research. So we performed the PCA on all the variables we decided to put in our final data analysis. And what you can see here is that all the variables, all the canopy variables I explained before, except the leaf area index, clump together into one direction of the PCA axis, and all of them load very high on this first axis. So we decided that this will be kind of a canopy heterogeneity index in further analysis. And what you can also see is that the Shannon index of the understory, the vertical connectivity, and the cover of the herb layer clump together also in the same direction and load very high on the second PCA axis. So the second PCA value was, for further analysis now, like kind of a diversity and structure index. And when you fit these on the count of singing Babetistes males, we have significant correlations, which means the higher, we have a significant correlation between higher canopy heterogeneity and a higher diversity in structure on each side and the higher count of singing males. And actually, canopy structure or canopy openness specifically has been linked to arthropod diversity in several groups through the last years. And that's why we asked ourselves if Babetistes sericata maybe could be an indicator organism for forest habitats that might promote diversity. This is a question I'm happy to discuss, maybe, because we all know that orthopterans have been very useful indicator organisms in open land habitats for quite some time now. And in the end, I'll give you an outlook on what we are doing right now. In cooperation with the remote sensing department of our university, we developed a habitat model with our presence data we collected throughout these last years. And we included several climate variables like annual precipitation amount, mean diurnal range, or isothermality. But we also included biotic variables like the canopy height or several vegetation indices. And what we will do in this summer, starting next week actually, again, we classified this model in several categories, like 3 of them, like low suitability, medium suitability, and high suitability. And again, we will do a random point survey on all these categories, kind of to test this habitat model approach. How does it work? How does this habitat modeling approach work? So thank you. And maybe there's time for some questions.
Thank you very much. We still have time for a few questions. Yeah, Anton?
Maybe something to
Yeah, that's a very good question because what we saw with the sound camera too is that when you have an ultrasound detector and you count one male, and you see in the ultrasound camera that there are two males like alternating, but still we thought, okay, when we can count with the ultrasound detector more than one or more than two or more than three, it's still a measure for, okay, there are more individuals than on the other plot where we can only hear one.
Another question to this topic. It is up to 50 meters, yeah?
15?
50.
50 the ultrasound detector?
Yes. Because you count the population as it up to 50 meter radius.
15.
15?
Yes.
Because I saw that.
50. Yeah, but we walked around in this area. Yeah, we walked around in this area. The range of the ultrasonic detector is, I guess, 20 meters maximum. Yes.
Ami.
I would like to know if you have also data. You showed that the singing activity is highest in late June, July, and so on, and the daily activity, you merged all data.
Yes.
Is there a change within the season that the highlights of singing during the day are switching from one time to the other?
Yes. Our data shows that it's this is not dependent on the time of the year, but more to the temperature. Like when it's in the middle of July or end of July, it's exceeding 32 or 33 degrees, they start later in the day. But, and when it's already a bit cooler. When you exclude the temperature, it's like throughout the year the same pattern.
One would expect that, of course, because temperature is related to the season, there's a change in the journal activity pattern.
Yeah, but the season is quite short in the end. Yeah, it's mainly July, August, and then there's also not such a big variation, and it's always a little bit matter of the kernel conditions, whether there's a heat wave in July or August. Yeah. Paulo.
For the relation with of the song with the temperature, we observed the same in Barbitistes Vicetinus. But I would like to ask, if I correctly understood, you found Barbitistes sericauda only in woods, because in my experience in northern Italy, it lives in clearings or in the prairie, in the edge with the wood. And I observed in many cases a movement not only from top to the ground of the wood, but from the wood to the prairie. It's very easy to find Barbitistes sericauda 50 meters, 100 meters far from wood habitat. In Germany, it's strictly related to woods.
No, it's not strictly related to woods. We have these observations in Germany too, but still in our project we focused on the forests, so just to find a pattern here in the forests. But we have these observations in Germany too. Not so often, but still.
Yeah, I mean, I think if we find specimens outside of forests, it's usually clear cuts, which are now overgrowing with about brambles or so bushes where there was formerly forest. Or I also have the impression that the further you go south, you found them outside the wood, because in southern Germany they're also sometimes in gardens, but in our region we never have them in gardens, only in the woodlands. Yeah. Sebastian last.
Yeah, I agree. In the high elevations in the south of Germany, in the Alps, it's also quite common outside of forest habitat.
Okay. Good. Okay, I think we have to go to the coffee break now. Until 11:30, okay?
Yes.
And the coffee break will be
Downstairs
in the garden. Good.
Thank you.
They behaved quite well, all were inside.
It's here. So if you want, just click.
This, so I have always to use. Do you have not such a?
Yes, but you know we are recording, so it's strange if you.
So I have to click here or what?
Yes, click. And punto coraggio.
Gaan we het zeggen.
It's the first one.
I have to click it twice.
Okay, here we go.
And then it's on. Off
If
for
you
the,
wanted to just ask the guy in the small room, will do it for you.
no, it's just because, I mean, you always have this one here, but you don't see how it's, yeah. Okay, and
mouse.
Mouse as a pointer.
Yeah. This works. Okay.
You
To make it a pointer.
yes, this one,
Okay.
the first one.
Puntatore laser. Okay. Makes sense. Okay.
Yes.
Good.
Because otherwise it will not be recorded, I think.
Or you can just use the.
Yeah.
If it works. Yes. I should bring a pen.
Good idea, yeah. Please take the seats. We will continue now. I know the coffee break is always the most important thing in all of the congresses. And I hope you had good discussions, but we will continue now with the next presentation by Anton Christin. He will talk about the expansion or recolonization of Agrida and Acrotylus in the northern range margin in Slovakia.
So this is nice surprise. I didn't expect that I will be the first in this section. So this story is about the expansion, spreading, distribution, colonization, recolonization are very frequent stories recently. And let's tell us something about these two genera, these two thermophilous genera, Acrida and Acrotylus at the northern range margin. Try. How to move page down? Okay, fine. What is the problem? So, how, Philippa, please, I cannot move. You
can move it like this.
And how to, because it was first. Here
if you want to go.
Only.
Okay.
Okay. Sorry for this technical problem. Okay. Everybody knows that in the last 100 years, the annual temperature increased mainly in Central Europe up to 2 degrees Celsius, and we expect continual increase of the temperature. And in many thermophilus, we expect also the range shifts. And then which kinds of habitats can be mostly influenced? Mostly the sandy habitats, and they belong to the warmest, and in Central Europe also the most endangered habitats. And they are inhabited by peripheral populations of some Mediterranean species like Acrida and Acrotylus or some other species. We put there two questions. One is in the title. If we can observe the expansion or spreading, really spreading northwards, or recolonization, so colonizing the former historical sites in these three thermophilous Mediterranean species in Central Europe. And the second question will be, these are the three species that you see, if could we colonize or recolonize even the disturbed and ruralized habitats. These sandy habitats mostly are such exploited sand mines and heavy, very extensive pasturing and so on. So we followed the changes in distribution within 160 years from the monograph of Przewalski, was published in 1867. Then we followed or studied the changes in habitats in three categories of disturbance, and then we checked also the changes in abundance and phenology of these three species in the area here shown some 800 kilometers, a little bit northeast from here. In this period, we checked more than 4,300 sites in all mapping squares in Slovakia. You can see the sandy sites mostly distributed in Slovakia. We checked within the sites 92 sandy sites, 44 in the western part of Slovakia, 48 in eastern part. Oliver already mentioned the sites of Gamsocles glabra. Then we used the sweeping nets, 500 Swiss per check per site. The minimal study site plot was 2 hectares. And then, this was in the field, and then we reviewed all published data since this monograph, Przewalski 1867, and we verified all previous data on all these three species. They are well-flying, very well-flying species, so expected to be good for the expansions. They are thermophilous. They are mainly on the northern border of Samophils. They are very abundant in core area of species range. Then what we found in previous paper also with international team, also Thomas is here from Austria in the European Journal of Entomology in 2003, is more every topic, this Acrida hungarica, than both Acrotylus species in the more northern border, which are more stenotopic. So distributional changes. We know this range from the last IUCN red list Acrida hungarica is distributed typical in Mediterranean up to Slovakia, and there are some questionable distributions in Czech Republic and northeastern Austria, and a very similar distribution like Acrida, a little bit known in Portugal, is Acrotylus insubricus, and within the Acrotylus insubricus range we can find the populations of Acrotylus longipes. Only sometimes we can find some inland populations, mostly they are coastal populations, and it is very interesting that we found the populations of Acrotylus also in Slovakia, mostly in this southeastern part, because in southwestern part we could not confirm the old historical data of Acrotylus species. So the previous paper, what I mentioned already, found in northwestern Pannonia, in these countries, Slovakia, Austria, Hungary, and Croatia, that in this mean July temperature between 18 and 20 degrees, that the Acrida hungarica has a huge horizontal increase of occurrences in these countries in the last 20 years. We can see these red circles. For example, in Austria, there were 3 former occurrences. Now there are 5, for example, and they confirm the old historical sites. Then in the northwestern Pannonia, this is much more. We had here in this circle 51, and here is 118, 22. So, and what is the reason? So, it seems to be that the increase the number of grid cells with hungarica, because we've studied Orthoptera assemblages in a lot of squares, was not correlated with more intensive sampling effort. So, by person correlation, we can see the blue is the research and the orange is the Acrida occurrence. So, not correlated, so it seems to be that so there are really expanding or spreading at least horizontal way. So what we found further, that the trend in effective heat sums more than 10 degrees in June, August by Mann-Kendall Turn test is significantly increasing. Like when we will do it, I think everywhere in Europe, we will find this trend. So, and what about this our study site in northernmost range margin in Central Europe, Slovakia? So, Acrida hungarica, we can see, we found, where is the density? Ah, sorry, there is missing something, but okay, I can explain. So, excuse. Okay, now we can see the red dots are the data from 2000, or after 1965, the black dots are before 65. It's interesting, this is missing some text here. So, but this is very rough data. You see the distribution, former black, this is historic, never more fine. What is, we can see only small northern distribution here in this. So, period up to 65, black, period after 65, a red one, but several of these are overlapped, the circles. Maximum density was 38 adults per 1,000 hectares. Maximum home range in the Danube area is 11 hectares per site. So, in 4 periods when we are checking this, we can see also, very nice, that the black, all sites, historical sites, are not reached still now. But from yesterday, I have some messages that they were found in somewhere here, nearby the Czech and Austrian border, but it's absolutely new. But what, they are increasing the data, but not crossing the northern historical range border. So, to Acrotylus insubricus, here we can see the black dots, they are the data up to 1965. We have 3 in Danube area, in southwestern Slovakia we have 2 from eastern, and now we have 8 dots after 65, but I should underline all the data are coming from 2019, and each year we have the populations, very nice populations there in southeastern Slovakia, what is connected with Gamsocles, Labra, and so on. Insubricus has maximum density 26 adults per 1,000 square meters, and maximum home range in eastern Slovakia is in this site, and we can find that no confirmation of historical data in Western Slovakia. Longipes is the most stenotrophic species only on fine sandy dunes. We can find the data only here, one historic data, and there are two new recent data after 2003, only from the last year and from this year. So, and then we can find also one very interesting finding of one specimen nearby the Polish border, but it was only one flying male with maximum density 11 adults per 1,000 hectares, the maximum home range 5 hectares per site. It was in this southeasternmost point. What about the habitat disturbance types and changes? The first are natural sand dunes, called like a natural. We can see, you can imagine how they see. Then traditional use sands and pastures, we called it a semi-natural, and the intensively used disturbed bare sands and grounds used very, very extensively, you can say, even for Insubricus is typical such quarries in the limestone areas. So when we are comparing the periods up to 1965 and after 2000, we can see in all three species that in former periods there were, here in brackets are the number of sites. In Acrida hungarica, also in Insubricus and Longipes, they occupy more completely disturbed or semi-natural habitats and less the completely natural habitats. To abundance and phylogeny, this is quite interesting. The Insubricus we found is frequently overwintering, and we can find the Insubricus from 16 February in the field up to 21st May, and then in autumn period from last decade of August to middle of November. In laboratory still, I was calling today my friend, in laboratory still we have 4 individuals living. From the previous year, they are maybe 300 days old, adults. Then Acrotellus longipes, you see, no one individual was overwintering in the field, this yellow, and we can find also the first individuals, adult individuals, on 21st of August or 25th of August, excuse me, and the last one, 3rd, 11. And then Acrida is well known from last decade of July as adult to October. So in the comparison with the old data from Barnabas Nagy from Hungary from 50s, 60s, the Longipes was, Insubricus was known only from last decade of April to first decade of May. Then we found, you see, the data increase in huge period nowadays, recently, and old data from Barnabas Nagy were from autumn, also for shorter period, and not so long to November, because the last October and November months, mostly in Hungary and Slovak area, was colder than today, and we can see in Longipes even shorter period. So there are some changes in seasonal distribution of this species. About the nymph occurrence, we confirm only the old data from Barnabas Nagy from 58. You can see all these nymphs, Longipes, Insubricus, and Hungarica are from our sites. So these are good breeding populations there. So to conclusions, recolonization, yes. Expansion northwards in these 3 species we cannot confirm, but all 3 species spread in new semi-natural and even disturbed habitats. So thank you for attention.
Thank you, Anton. One question? Yeah.
Could it be that Barnabas Nagy was not searching for grasshoppers in disturbed areas in the early days? Is there any bias in data? Maybe
Yeah, sure, it is. It is, because we cannot confirm in historical data how quality was the habitats, how deep was the disturbance of the areas. And this is always the question, the categorization of disturbance for particular habitats. And mainly the sandy habitats, stony, gravel pits, and so on, They are sometimes very complicated but what we compared from our Slovak area, that they were very good data about the quantification and qualification of disturbancy, because we knew that there were only the natural areas in that time where are now the sand pits and quarries and so on.
Okay, thank you, Anton. I think we have to move on. So next presentation will be by Andreas Hildpold on high importance of extensively managed grasslands for the conservation of Orthoptera diversity in agricultural landscapes.
Okay, good morning. So the study I'll talk about is about the value of grasslands from nature conservation, and specifically what kind of grassland is of high value. This data I show is more a side product of a larger project, which is the biodiversity monitoring of South Tyrol. South Tyrol, for those who don't know it, Südtirol in German is just north of it. Today in the morning I just took the normal morning train as all the days and went to Rovereto instead to Bolzano. It's a small country, 500,000 of people and 7,400 square kilometers, part of Italy, of course. So there we have in 2019 we started a larger monitoring program, and you see here that we have many monitoring sites all over the province with many categories, including running waters and forests and so on. And we chose the sites within a certain habitat type and mostly randomly, both in protected and non-protected areas. And the data I showed today is only from this. No, how is it going? From these three general habitat types. It's from grasslands, of course. It's from apple orchards, and it's vineyards and settlements. And so the sampling would rather look like this for this study. We have many different sites all over so 150 single sites with out of 7 land use categories and here today we show only the lowland and mountain data. Sorry, there is a problem now with the presentation.
It's stuck.
Stuck. The PowerPoint seems to be stuck. No reaction here.
Just wait. He will manage from the small one. If you have some questions.
Okay. Now it's restarting. Okay. Let's hope it's working now. Maybe I was just too fast. So here, glimpse into some sites we have actually surveyed, and of course for a monitoring program we have a look to many different taxonomic groups, and among others, of course, grasshoppers. I think they are generally a good indicator taxon for the quality of landscapes and habitats, and we also have the available expertise in the province. So the specific research questions we tackled within this analysis were mostly what is the role of the extensively managed grassland for the conservation of grasshoppers in comparison also to the more intensively managed agricultural habitats and settlements, and what kind of grasshopper community do we actually find in the urban and disturbed habitats, and finally also how effective is the agri-environmental schemes of the province for conserving grasshoppers. And about agri-environmental schemes, we have in South Tyrol a scheme which is called Landschaftspflegeprämien, and farmers get subsidies for especially for managing different kind of grasslands, mainly hay meadows, and they go up to 600 euro per hectare. And so in our site selection, half of the meadows were part of this subsidizing system. The methodology we have is adapted to the botanical survey, and we have plot sizes of 100 square meters with smaller transects with 50-meter transects in settlements, and we try to get all the species we find or which actually we may live in this patch, and the survey is being done in late August or in August. Here's some data of this study. You see that we have, after this, with this 150 sites and more or less 20 replicates per habitat, we have already reached a very good plateau for the single habitats, and we can already, it may not improve substantially by adding additional sites. And what we see here is the different habitat types. So the two on the left are two grassland types. There's extensive pastures and extensive meadows with the highest species, in this case it's species diversity. The intensive meadows are a little bit poorer, and all the other habitats are much poorer in species diversity, and the same is true for the abundance and the species richness. The extensive grasslands, both the extensive hay meadows and the extensive pastures are by far the richest habitat types, and the poorest habitat types are apple orchards, the intensive apple orchards you find in South Tyrol quite commonly, and settlements, and vineyards, corn and cereal fields, and intensively managed meadows range kind of in between. Looking at the community here, ordination, we see that it looks a bit messy. We see that there is some pattern, but I try to go a little bit more into detail into the following slides. So here we see that on the right side, we have all the more disturbed and more intensively managed habitat types together, which are vineyards, apple orchards, and settlements and so on, and they are relatively similar, share a few species, of course, like Anacridium, like Cortipus brunnius or Ruspolia, and they are quite generalist species in our area. The vineyards, a part of the vineyard is have a similar community than part of the pastures. It's mainly similar to the dry grasslands, which is because of these species, mainly Calyptomus siciliae, Homocestus rufipes, and Mantis religiosa. The intensively or semi-intensively managed meadows are here in both. We see that they are characterized relatively poorly by species like Pseudocarpus parallelus or Russelliana, and share, of course, species with the disturbed and the grassland And here to the left side, you see all the different grassland types with many, many more species and many species characterizing them. And when we have a look at the pastures in comparison to the hay meadows, we see, of course, that there is also a difference, mainly also ensiferous species, which mainly occur in the pastures because there are single shrubs and trees as well, like Barbitistes or Leptophyes albovitata. And also in the drier grasslands, we find also dry grassland specialist species. We see also that the species numbers between the different grassland types, comparing the mountain pastures and the lowland pastures, that the mountain pastures are actually the species richest habitat type of South Tyrol, richer than the lowland pastures, the collinar pastures. But when we look at the community, we see that they are quite unique, the colline pastures, which is due to a few species like Omocestus petreus or Stenobotrus nigromaculatus and so on. Looking at the red list categories, this analysis is not totally finished, but we also see that this same habitats I showed you before, the extensive hay meadows, the extensive pastures, they also harbor the largest quantity of red list species. Of course, here are also other habitat categories. The wetlands are those with most red list species. So I come to the conclusions. The extensively managed grasslands, both pastures and meadows, have a high conservation value. The subsidizing this extensive grasslands is of very high importance for Orthoptera and fauna, and our agro-environmental schemes seem to be very effective to do that. And we also recommend to improve the conservation of extensive pastures which are not subsidized. And for the more intensive land use types, they have in general a small conservation value for grasshoppers, and there is large room for improvement, especially also in urban areas. With this, I want to thank the whole team of the biodiversity monitoring, and I want to thank you for your attention.
Thank you very much. We have time for questions.
Not in the biomass, but in the abundance. And the abundance, of course, you could calculate the biomass if you wanted, yeah.
So you have a higher abundance in this
yes,
natural...
I may go back to that if there is time to this. This one. Here to the left, you see the abundance, and the abundance is higher in this extensively managed grasslands. And it's very, very low in the apple orchards, go down to zero, and also in the settlements, it's quite hard to find specimens. I have to say that we didn't use ultrasonic devices, which could be very useful, of course, for settlements and also for maybe not for apple orchards. I don't think that we would find many more species there. Yeah.
Okay, if there's no other question, I would suggest we continue with the next presentation by Ricardo Marinho Perez. He is actually a Mexican citizen. And so we will look over the ocean now and have a look at very special groups of insects, particularly the Tanaotseriidae, which is an endemic, endemic to Mexico and southern US, yes. Right? Yeah, and also the Xyronotidae. So some two very special groups, species poor groups, but very unique.
Thank you. So having a break from Europe, just a little bit because I'm going to talk a little bit what I've been doing in the last 2-3 years. Thank you to the support of the IUCN. So first, these groups are considering EDGE, that is called Evolutionary Distinct and Global Endangered Species, or groups. So these are groups very, very particular that you know, are unique evolutionary lineages, and Tanauseridae and Cyronotide fell into that category. Can remember the year, probably 2-3 years ago, the IUCN Grasshopper Specialist Group received funding for doing this project. And just a quick introduction, the desert longhorn grasshoppers are endemic to the California and Nevada in the United States, and in Mexico in the states of Baja California and probably Sonora. These grasshoppers break the typical rule that all of us learned that Celifera always have antennas shorter than the body. These are the unique group that break that rule, so now you know, or maybe you already knew, but that's a very, very important thing to know. And they are really rare, and the biology is not well known, and they do some kind of acoustic communication that is still not known. Here you can see the iNaturalist page, where you can see that these grasshoppers are very, very localized only in early spring, late summer and, late winter and early spring. So that's the reason probably that they haven't been studied a lot, because no one goes to collect in those places that early. However, is where they are really, really abundant. The peak of abundance is in March. The second group is called razorback bush hoppers, and this is also a unique lineage of 4 species in 2 genera, and there are unique and considered relic lineage. The sister group of these grasshoppers is the Trigonopteridae, that maybe you know from Southeast Asia. So it's really interesting, that story. And again, these are Paolo Fontana told me that they do some kind of stridulatory communication, but that's one of the research lines that we want to continue. And these grasshoppers are more, they are found in the tropics, and they behave more like the normal grasshoppers that we collect in from July to September, October, the typical field season in Mexico. Just for your quick information, well, you can see those two. Oh, yeah, you can see these 2 lines. This is where these families are. So, Cyronotide, as you can see, the sister group is Trigonopteridae from Southeast Asia, and Tanauseridae is here, no? Is together with this. So the current status, I visit the different museums in the United States where the great majority of the specimens are held. And what were the challenges that we have to do? We revisit the type localities at the same time that where they were collected, and at different time. We find new localities, and also we had a photographic record and video of alive specimens, and we collect the DNA, well, the samples for DNA. And we are in the process of do the IUCN Red List assessments for the 4 species of Cyronotide and the 3 of Tanauseridae, as is listed in the strategic plan of the ECSC. Bless you. In March 2022, I visited the California Academy of Science, that has a fantastic collection of Nearctic grasshoppers, particularly due to the proximity to all the area of California and Baja California. And I review and curate all the non-type material of Tanauseridae, as you can see. And after that, we conduct an expedition in California and Nevada. This is the typical habitat there. They are really rare to find in the day. They are most actively at night. And for instance, at night, we find them here. But when you return later or the next day, you cannot find them during the day. And it's quite interesting to know where they go, because as you can see, the vegetation is really scarce. I'm not sure if this is a video. Maybe. So you just see how they behave. You can see the male and the female there. And then the male is going to move the legs. So the next step is to have the right equipment to try to record this sound, because right now it was just an exploratory trip. But we were really happy to find them, because it looked like it was rare, but no, there are just there is that no one goes to collect them. And well, here are some of the pictures. We also recording what type of plants they were. Some of them were really spiny, as you can see there, here. And as you can see also, sometimes they are found just in the ground next to the plants, and you can see how they just get lost in the ground. This is a video again. These females were found during the day, as you can see where they are found, in this crazy place that is just like 20 kilometers from Las Vegas. And it's interesting because here, people, you know, crazy Americans, they used to, you know, they enjoy to shoot guns. So in this place, they just put bottles and shot the bottles. So it's full of glass and broken material, and it's where I found these guys. So probably this is the first case to mention that these grasshoppers are endangered to guns from Americans. So that's so crazy. I cannot say this in America, so I'm happy. I need to travel all the way here to say this. So
Remember that they are listening. We make the latest for the recording.
And it's crazy because this is just like, as I say, 20 kilometers from the horrible, useless city in the world that is Las Vegas. And I mean, I was happy to be there, because this is how the habitat is there. And fortunately, we were able to collect different specimens from the DNA analysis. This is the other species, the Mojavacris timberlakei. Again, these species are really localized, and this was March, and it was really cold. You will never imagine that you're going to find something, and they are there, so it's crazy. It's crazy because you cannot find any other species of grasshoppers. And again, these are really abundant during the night. And here are some of the pictures of this, and we found them in a place that was just recently born, so that was really interesting. Hmm? What? In which months? This was March. It's crazy. Only March, probably April, and that's all. You never saw them again. So this is the trip that we conduct. We start in Los Angeles, and then just do we did a loop to near Las Vegas, and then we return. We cross the Mojave Desert, the Death Valley, and that's the trip we did. The second part was an expedition now to find Cyronotidae in the southern Mexico. We follow the localities in the University of Michigan, used to collect a lot there, so they have all these digitalized diaries, and we went there and we locally find them. So this is how we find them now. And as you can see, this is a completely different habitat, but they are not associated in the typical place where you are going to look for grasshoppers. This is more like a forest grasshoppers, as you can see, they jump. So la alejas así. Locally, a rich Mexican that loves butterflies purchased a huge area in Chiapas state in southern Mexico. So this is where they are found. So I consider for the moment they are kind of not endangered. The local people know them. We couldn't find them. We showed to a local people, hey, have you seen this? Oh, yes, it's here. And he took us and we find them. And you can see how they are there. It's just that you need to pay attention. This is the species. And they have different morphs. So the next stage in our study is also to study the different morphs that they have, because some are like that and others are brownish, others have like white marks that resemble lichens. So it's incredible the diversity of this group. So this is part of the expedition team, and you can see here, or you cannot see here, the Cyronotide. Oh. Well, this is the other morph that I told you that now this kind of resemble, or confused between the white pebbles and lichens. So, but it's the same species, and they are just there like this. They are just next to the other. So it's just incredible. Interestingly, these grasshoppers are also really abundant during the night, but during the day, because it's cloudy and it's kind of dark in the forest and in the edge of the forest, you can also find them. Finally, we visit another locality in Oaxaca, and here we found another species from a different genus. But you can see that this guy just resembles the other guy there, or from the other species in other mountain range. So it's just super interesting. And again, well, a convergence. And again, here you can see other ones. Maybe it doesn't look that much, but they are kind of greenish because here there is a lot of moss. And this is another. This particular place is well taken care from the locality. In fact, when you just stop the car and try to collect, in 10 minutes people appear. What are you doing here? So we ask for permission, and they allow us to do that. And there is, in a certain special Mexican government treat, that the Mexican government pay for keep the forest due to the services that they provide. So this was the Cyronotide expedition that we conducted that time. Punta Torre Láser. So these are the places, and this is the place in Oaxaca, and you can see the first species is in a different mountain range in Chiapas. So we try to collect here, but there is nothing here, so something is going on there. After that, I visit the Academy of Science in Philadelphia, that if you've been there, is probably the best Orthoptera collection in the world from material from the Americas. And there I photograph and review the non-type material and photograph the type material that was housed there. So all the type material is already digitized. And from a previous grant, the holotype of Cyronotus azteco is in Geneva, so I was able to photograph it there, that type in 2016. And I just finished a postdoctoral fellowship in University of Michigan that has the second most important Orthoptera collection from the New World. And then all the material of Cyronotide is housed there. So I was able to photograph there and obtain all these images. And just like 2 weeks ago, I curated the material that was recently collected in UNAM in Mexico. And now we are starting to conduct the DNA analysis. We are just missing the Thanosauridea from Mexico, but we are going to get it soon. So the most important is that we review the type of non-type material from the collections. We visit the legacy localities and new localities, and we find them. And the great majority of the localities are under some time of protection. So now that we are going to conduct the assessments, I'm positive that the assessments is not going to be something like data deficient, but it's going to be a more elaborated one. In Metaleptia, the newsletter of the Orthoptera Society, I share the 2 expeditions in these articles. So regarding the challenges, the ones in red are complete, and now we are just working in the assessments and the phylogenies, and of course, I know here there is a lot of colleagues that study sound, so I will be interested to talk of how can we try to record some sound from these groups. So I want to acknowledge these institutions and people that helped me to conduct my research. So thank you.
Thank you, Ricardo. Very interesting species, I think. Are there questions? Yeah, José.
Yeah. There are localities but I wasn't able to, the stupid Americans didn't allow me as Mexican to cross the border to Mexico. But and of course, there are endemic, because when they say they are not endemic to Mexico, well, there are so endemic to the region that basically they are endemic. It's just a political thing. But we are very close to gather the material from there, so we can do the phylogeny.
Okay, good. Thank you. There's another question, sorry.
Well, no yet. We are in the progress of that, but for the Tanauseridae, because there is very, very few vegetation, they are almost in the plants that are just there.
Yeah, but you found a species of plants.
No yet. We are exactly, a botanist is helping us, but apparently there are certain correlation, because you can see even the form of some Tanauseridae, they are like resemble certain branches of the probably species of plants that are associated with.
Okay, so we will move on to the next presentation. This one will not be recorded and streamed. Yeah, so this is why it was moved to the end of this session by Armin Landmann. You may know Armin. He's a very well-known Austrian orthopterist who's doing a lot of research and has a lot of diversity in Tyrol, I remember. So we will hear something about the, to the new red list of the Austrian grasshoppers. He is shown here, but I think that will be mentioned.